Today we are starting new chapter biomolecules and we will be doing NCERT exercise question 10.1 to 10.5. First question is, what are monosaccharides? Monosaccharide term we can break into mono means one and saccharide is derived from a term saccharin which means sugar. So all those carbohydrates which contain single sugar molecule, they are called as monosaccharides, and these monosaccharides, they cannot be hydrolyzed to smaller molecule. This is their general formula where N stand for 3 to 7 and naturally we have 20 types of monosaccharides. Now these monosaccharides are classified on different bases. First on the basis of functional group, and they are also classified on the basis of number of carbon atom. So on the basis of functional group, they are classified as aldose and ketose. Aldose are those monosaccharides which contain aldehyde group as a functional group, while ketones are those which contain ketonic group as a functional group. In addition to this, they are further classified on the basis of number of carbon atoms. So triose is the sugar containing 3 carbon atoms, tetrose with 4, pentose with 5, hexose with 6, and haptose is containing 7 carbon atom. We'll stop it here. Why? Because we have limited this number from 3 to 7 in the general formula. Now here are a few examples. The first one is labeled as aldotriose, second one as aldohexose, and third one is ketohexose. Now we will understand why we are classifying them in this way. First part, aldo means containing aldehyde group. Therefore, in this molecule we have aldehyde group, and triose means there are 3 carbon atoms. So here we are having 1, 2, 3 carbon atom. Okay, so therefore it is called as aldotriose, and name of compound is glyceraldehyde. Second one is aldohexose. Aldo again means aldehyde group, and hexose means maximum 6 carbon atom, and therefore it is called as aldohexose, and this compound is glucose. Third one is ketohexose. Keto means ketonic group present as a functional group, and here second carbon is the ketonic carbon. Hexose means 6 carbon atoms are present in the molecule. So this is all about monosaccharides. Next question, what are reducing sugar? Now sugars are of two types, reducing and non-reducing. So what are reducing one? All those sugars or carbohydrate which reduce Fehling solution or which reduce Tollene's reagent are called as reducing sugar. Okay. And you have to remember that all monosaccharides are reducing in nature. Two examples are glucose and fructose. Now we will see that in lab, if we are doing this test for reducing sugar, how we will identify that our giving sample of sugar is reducing or non-reducing. Okay. So first one, we are treating sugar with Fehling solution. In lab, Fehling solution is in the form of Fehlings A and B. So first we have to mix these two solutions, then we have to treat it with the sugar solution, and after heating in a water bath, we will be getting a reddish brown precipitate if your sugar is reducing. Okay, and why it is happening? Your Fehling solution is basically a solution of copper ion, Cu2 plus ion, and when we are adding sugar into this and then we heat this solution, we will be getting a reddish brown precipitate, and this is the precipitate of copper oxide, CuO. So in this case, your Fehling solution has been reduced from copper 2 plus ion to copper, here we are having Cu plus ion in the form of CuO. So this process is what? Reduction. Second one, and the color changes from blue to reddish brown. Second one is Tollene's reagent. Tollene's reagent is basically a solution of silver ion, that is Ag plus ion. And when this Tollene's reagent is treated with sugar, and we heat in a water bath. So what change we observe? We are getting a silver mirror deposition on the margins or edges of the test tube, or on the inner lining of the test tube. Okay, so why we are having this silver mirror formation? This is nothing but silver metal. So here your Ag plus ion is undergoing reduction to Ag, and this has been done with the help of the reducing sugar. So again, our sample is a reducing sugar because it is capable of reducing your Toluene's reagent when we treat Toluene's reagent with this one. So this is all about reducing sugars. Next question. Write two main functions of carbohydrate in plants. In plants, carbohydrate play two roles. First role is as a structural material, and the second role is in the form of reserve food. Okay, that is food storage. So polysaccharides, cellulose, act as a chief structural material of plant cell wall. Okay, that means plant cell wall is made up of cellulose or polysaccharide cellulose. Second point, the polysaccharide starch is major reserve food material in plants. Okay, so food reserve is in the form of what? In the form of starch, which is what? A reserve food material for the tiny plant in the form of seed. So this reserve food is basically stored in seed. So these are two main functions of carbohydrate in plant. That is, first to act as a structural material, and second one is as a food storage. Next question. Classify the following into monosaccharides and disaccharide. Here they have given a list of compound and we have to classify them. So out of these, monosaccharide means which are made up of single sugar molecule. Monosaccharides are, the examples are ribose, 2-deoxyribose, galactose and fructose. Okay, and the disaccharides are means which are made up of two sugar molecule. So they are maltose, and that is malt sugar, and lactose, the sugar which is present in milk. So this is all about different types of your carbohydrate. Next question is, what do you understand by term glycosidic linkage? Okay, so what is exactly glycosidic linkage? The oxygen linkage through which two monosaccharides are joined together to form a molecule of disaccharide is called as glycosidic linkage. Okay, so here I have shown you a molecule of maltose, which is what? Disaccharide, and it is made up of 2 alpha D glucose units. Okay, and these 2 alpha D glucose unit are connected by a oxygen bond, and between first carbon and the fourth carbon of first molecule and the second molecule respectively. And this linkage, that is glycosidic linkage, is somewhat like the linkage which we find in case of ethers. These are the compound. Ethers are having this type of bond, that is carbon oxygen bond. The similar type of bond we have in case of disaccharide. So here this linkage, that is oxygen linkage, is called as glycosidic linkage or glycosidic bond, and this bond always here exists between the first carbon of first molecule and the fourth carbon of the second molecule. Okay, this is all about glycosidic linkage. We'll